Topic 7, the cytoskeleton and molecular motors. At this point, we've talked about a whole bunch of things. We had some introductory stuff. We've talked about some different organelles and different cell structures. This kind of fits into that category. Sometimes I squeeze it in before the midterm. Sometimes I squeeze it in after the midterm. It also is a good kind of transition to the next part of the course where we're going to talk about energy. So we're going to talk about enzymes and respiration and photosynthesis and all that. So let's talk about the cytoskeleton. The cytoskeleton is good for great pictures. Look at all this fluorescence microscopy. You can see these wonderful little fibers and structures all over the cells. And that's exactly what they are. The cytoskeleton is fibers, and they are proteinaceous, meaning they're made out of proteins. You can't usually see them under a normal light microscope, but you can see them very well under fluorescence because we actually have some great stains for studying the cytoskeleton. And you can see, for example, in this picture here, some wonderful colors, and the different parts of the cytoskeleton get labeled with different colors. So there's a more of a cartoon of the cytoskeleton. You can see the three components. We have the microfilaments, intermediate filaments, and microtubules. This is the worst part of the cytoskeleton, the names. Microtubule, microfilament, intermediate filament, they all sort of start to sound the same, and you don't really know anything about them. So hopefully by the end of this lecture, you will know a little bit about them and have an idea of what they're all about. Here's another picture of the cytoskeleton in action, and something all of these components have in common is that they're involved in cell shape and support. So really like a skeleton, meaning they're kind of strong and giving support to different structures. So this is microvilli. You probably have heard of microvilli, and I think Biology 30 is where you talk about them. So these are found in your intestinal cells, and what they are doing is giving your intestinal cells more surface area for the digestion of your food. So I've kind of already said this. This is a bunch of fibers. They're proteinaceous, and they're all over the place. Cytoplasm is kind of the main part, although there's some we've already talked about that are found in the cell nucleus. So the nuclear lamina are part of the cytoskeleton. So here are the three components of the cytoskeleton. And I'm going to make a table in a couple of minutes, and we'll slowly add to it. The big ones are the microtubules. The smallest ones are called the microfilaments, or sometimes we call them actin filaments. And then the ones in the middle are called intermediate filaments. So hopefully that one's obvious that they are in the middle. And they're all different structures made out of different protein components. So another thing to notice, you may notice a lot of these pictures that have the cytoskeletons are often using the same colors, and that's because we have some great stains for fluorescent microscopy. I like this picture here because you can also see the different components of the cytoskeleton are found in different cell regions. So for example, the microtubules there are found throughout the cytoplasm. The microfilaments are kind of found closer to the membrane, closer to the cell surface. So let's take a look at these, and you're going to be looking for these components in this week's lab exercise as well tomorrow. So there are the subunits, tubulin, actin, and keratin. So we'll get to each of these. Like I said, I'm going to talk about each of these in a bit of detail. So let's talk about these microtubules. So I'm extending the word tubules to kind of emphasize that these are tubes. So hopefully that will help you remember the name, tubules. Micro because they're small. The proteins are called tubulin. So anytime you see IN on the end of something, it's often a protein like insulin, tubulin, flagellin. We're going to talk about flagellin today. You can see these are largish. They're the largest ones. They're 25 nanometers in diameter and involved in a whole bunch of things which we're going to talk about. Here's the microfilaments. We'll come back to them. They're made out of actin and intermediate filaments. You can see the different sizes there. So let's start off with our notes. And like I said, I have going to make some notes here. And we're going to talk about the three different types of cytoskeletal proteins. You can see what I've done is I've started making a table. I have the three components, and I'm going to kind of fill in some details here. So if you miss anything, don't worry, we will come back to this. But we can start here and talk about microtubules are made of tubulin. And Notice I have it in green. I guess my green font didn't show up very well, but it's green because they usually stay in green. So, and I'll put that these are the largest. So these are about 25 nanometers, if you care about the size. And the locations, I'm going to put cytoplasm. So I will come back to this. If you didn't get all that, don't worry, I will move back to this shortly. Let's talk about some of the functions of the microtubules. So like I said, they're the largest, and they're made of tubulin. And you can see tubulin is a dimer. There's actually alpha tubulin and beta tubulin. There's even a gamma tubulin. It doesn't really matter for the sake of this course, but they are proteins that make up these tubes. So the tubulin, maybe you can see from this diagram, I meant to find a little bit of a better one. They kind of coil around each other and make a hollow tube. There's nothing on the inside of the tube, at least that we know of, maybe a little bit of water. So nothing is going through them. They're not like straws. This is just how they assemble and make their structure, and 25 nanometers. So where have you seen microtubules before? You may have seen microtubules before if you've heard of these things, centrioles and centrosomes. We're going to talk about centrosomes and centrioles in a bit more detail in topic 13 when we get to cell division. You probably know they have something to do with cell division. And so, by the way, you can see in this diagram here, right, that we have two centrioles, so 1 and 2, and 2 centrioles make a centrosome, right? So that's just kind of the way the terminology works. So you can think of a centriole as kind of like a starting point, an anchor for the microtubules. And like I said, you're probably thinking, okay, this is something to do with cell division. So another name for microtubules are actually spindle fibers, although that name is only really used when they are used in cell division. So you have seen them before, they're just maybe with a different name. So there's some electron micrograph of some centrioles. If you look carefully, you can see it's a bunch of little tubulins, and you can see in that cross section, you've got these kind of triplets of microtubules, and then those triplets are put together in a larger structure. So there's a picture showing those microtubules that are extending from the centrioles, and they're grabbing the chromosomes. They grab the chromosomes at the centromere. So there's a bunch of words that we're going to talk about when we get to cell division. So centrosome, centriole, centromere. If you're not familiar with them, don't worry, we will get to them, and we'll write some definitions when we get to topic 13. So you can see that the microtubules are growing out of the centrioles, and they're grabbing the chromosomes and separating the chromosomes during mitosis and meiosis. So there's one function of the microtubules. And like I said, I'll get back to that Word document in a minute and fill that in. I wanted to show you this, this nice little video. So what you're looking at here is actually 3 cells. And you could probably see the big dark things in the middle, like right there. That's a nucleus. And what you're seeing is somebody, I'm not exactly sure how this technique works, but somehow they label the end of the microtubules. And the microtubules are growing or shrinking. We'll replay that. They're growing or shrinking in the cytoplasm. So these microtubules are pretty active things. They're not just moving around chromosomes. They're actually moving around all sorts of things in the cell. So all those organelles we were talking about before, we have those little vesicles. They're made in the Golgi, and those vesicles will, they're made in the endoplasmic reticulum. They're moving to the Golgi, and then they move from the Golgi to somewhere else. And that's actually what we're observing here. And in fact, this is in real time. You can see it very, very quickly. So maybe I'll come back to that in a second. Let's just go back to our Word document, and I'll fill in a couple of those things. So functions, we have spindle fibers. So this is chromosome movement during cell division. Number 2, we have organelle movement. And we're going to talk about flagella. And cilia in a moment here. Okay, so let's go back to that PowerPoint. I know you guys saw the next slide here. So I want to talk about the roles of microtubules in flagella, and cilia. So we've already talked about flagella of one type, the bacterial flagella. And if you remember the bacterial flagella, they rotate and they go clockwise and counterclockwise. So this is a very, very different structure. It has the same name, kind of, does the same thing, helps them swim or move, but it doesn't work in the same way. So you can see in that diagram there's a sperm cell, and of course flagella in a sperm cell are kind of like these big tails. They're big floppy things and they move back and forth. And that's actually how flagella work. There's another structure here that's very, very similar. You can see on the bottom part, they're called cilia. One is cilium. And they're very, very similar. They're just smaller. So a cell may have one or maybe a couple of flagella. Cilia, you're looking at hundreds of them. And they're more like a bunch of little fingers. And the little fingers sort of paddle and they sort of move back and forth like tiny little tails or tiny little floppy paddles. I'll show you some videos. I want to talk about these structures. So there's a couple of videos there. You can see there is on the top, that's a mouse sperm, I believe that video said, and the flagella is kind of flapping back and forth and pushing it through the medium. The diagram or the video on the bottom is cilia on a paramecium, and they kind of look like little hairs around the edge of the organism. And like I said, you can picture it as just hundreds of tiny little paddles sort of flapping back and forth, and it's sort of in a little bit of a figure-8 motion is how they do. They have a power stroke and a slow recovery stroke, just like what you do with a paddle. So you're going to be taking a look at these structures in tomorrow's lab exercise with some videos I've provided for you. Here's a couple other videos. The one on the top is the euglena. So euglena is one of the organisms we normally look at in lab 5, and you can see it has a flagellum. And the flagellum, it just has a single flagellum. It's a very interesting organism because it's also photosynthetic. On the bottom, we have a paramecium. Again, just like the previous slide, you can see how it's moving around, and it's kind of funny how it twists back and forth. I think that's really interesting. They're very, very fun to watch, which is unfortunate we're not getting to look at them in person this semester. So by the way, you probably know that there are some human cells, so sperm cells that have flagella. There are human cells that have cilia as well, and cilia are found down your respiratory tract. So they push mucus is what they do. So rather than moving the cell, they're moving mucus. And so when you get sick, you detect a lot more of that mucus usually, and it's brought up the throat. But that's part of one of our defense mechanisms as bacteria or viruses, go into our lungs. We're trying to push them out amongst the mucus. So here's the structure of the flagellum and the cilia. And I want to talk about it a little bit, and I'm going to draw a picture for you in a minute. But this blue thing here, that's the cell membrane. So this is very different from a bacterial cell. It has a cell membrane around the flagella. It has all these microtubules, and just want to point out those little red things. Those are something called motor proteins. So notice this structure here we call the 9+2 arrangement, because you got 1, 2, 3, 4, 5, 6, 7, 8, 9 doublets all around in a ring, and there's a second doublet in the middle. I'm not sure why 9, but I guess our cells have decided that's a strong structure. And there's an electron micrograph. You can see that 9+2 arrangement is very, very nice. So here's kind of another diagram. I like this one because you can see the membrane kind of has a little bit of a fold to it as it's in that anchoring area. The anchoring area is called the basal body, by the way. And so I do want to, like I said, draw a diagram for you and compare these two structures. But I showed you, I think, the same slide before, and just to emphasize how different these structures are between prokaryotic and eukaryotic flagella. So I wanted to show you this little video. These are some sperm cells, and notice my note there. I'm telling you that these are powered by ATP, right? So you probably know what ATP is a little bit. It's found in the cytoplasm. So these flagella are continuous with the cytoplasm. They're surrounded by the membrane. So what this person is doing is they have a little instrument, and they're going to cut off the flagellum of one of the sperm cells. So let's just watch what happens here. So here we go, it gets trimmed. And notice it keeps moving. So it will keep moving as long as there is ATP there. If this was a bacterial flagella, there's nothing keeping it powered. It would stop moving. So I just want to talk about those motor proteins, and then I'm going to draw you a little picture. These motor proteins are called dynein. There's actually different types of motor proteins. What do I mean by a motor protein? I mean it's a protein that uses ATP and causes some sort of movement. So I think on the next slide it shows the movement in a little bit more detail. Yes, it does. And so you can see you have these two doublets of microtubules. So I'm looking right here. And the two doublets have these little dynein proteins, and they're almost, I sort of think of them as tiny little legs. They ratchet back and forth. They just move back and forth with ATP. So you can see ATP is going in there, it gets hydrolyzed, and it causes one strand to kind of, they kind of slide like this along each other, back and forth. So they're a little more floppy, so they kind of flop back and forth like that, and that allows the motion of the flagellum or the cilia. So let me just take a look here. I have one other motor protein I'll show you, and then I want to draw a diagram. There's another motor protein. You can see it's moving a vesicle along one of those microtubules. Okay, so maybe first we'll go back here to our notes. Oh, I've already put flagella and cilia on there. I'm not going to write anything about microfilaments yet. Like I said, what I want to do is draw a couple diagrams for you and try out this laptop pen I have, and hope I can draw nicely for everybody. So here goes. We're going to draw a structure, just very basic. Don't need to be too artistic, of a bacterial flagellum. So here goes. A bacterial flagellum has a bunch of proteins that anchor it into the cell membrane and cell wall, and that's actually also called a basal body. Write that down. Basal body. Okay, so what I'm going to do here is I'm going to draw for you, so that there is the plasma membrane. So PM for plasma membrane. We'll make this a gram-negative organism, so we'll put in a little bit of peptidoglycan there. So PG for peptidoglycan. If this is a gram-negative organism, it is going to have an outer membrane as well, so OM for outer membrane. Okay, so it's anchored in the membrane. That basal body is also a bit of a motor. This is something that can rotate. And there's another structure here, and believe it or not, they're called hook proteins. And this structure here is called the hook. So there's a bit of a bend to it. If it was just straight, right, you could imagine it's not going to propel the organism through the media. So it's got to have a little bit of a hook to it to give it some propulsion. And then we have this big long part, which I really don't have enough space on here to draw, but I'll draw some of it. And let me just maybe get a different color. So this is this is made of something called flagellin proteins. And that's the big part of the flagella. I don't know if it has a special name other than flagellum. And that's made of flagellum proteins. So a couple other notes about this, right? Something that we've talked about already before is that this thing, the motion, is it rotates. So it rotates clockwise and counterclockwise. So that's CCW for counterclockwise. So there we go. If I could make my drawing move, I would do that. And the last note I'm just going to put down here in the corner is, what is the power source for this? So this is powered by something called the proton motive force. So let me just fix that there. Powered by the proton motive force. So you're probably thinking, okay, what is the PMF? What is the proton motive force? We haven't talked about it yet. We're going to talk about when we get to respiration. But you can probably imagine it's involving protons. So protons, of course, are hydrogen ions. And they're making some sort of force that has to do with the membrane and a gradient across that membrane. So more on that when we get to respiration in a couple of units from now. So there's my bacterioflagellum. Hopefully you like my diagram, and it gives you kind of a basics of how large it is. I guess the last thing to tell you is about how big it is. So the length is about maybe 2 to 5 micrometers. 2 to 5 micrometers. So a bacterial cell, like something like E. coli, is maybe 2 to 3 micrometers. So the flagella is about the same length as a cell, and sometimes a little bit longer, in general anyway. Okay, so I'm going to go, and I don't have enough space here. Usually I have a nice big whiteboard, and I draw one on one chunk and one on another chunk. I'm going to draw you a picture of the eukaryotic flagellum now. I'm just going to grab myself a new digital whiteboard here. So here goes. Eukaryotic flagellum. Okay, so we're going to start exactly where we started last time. I'm going to make a basal body for you. Although this basal body here is made out of mostly microtubules, so it's a very different structure and massively larger. So there's my basal body. And okay, so here goes. Again, just excuse my messiness. I'm trying to be as neat as possible and drawing this thing as, you know, I'm not an artist. I'll just say that. So what did I just draw here? This is the plasma membrane. Okay, so notice the plasma membrane is going all around the flagellum this time. Okay, now what I'm going to attempt to do, and maybe I will draw in the same color here, I'll draw it in the black. I'm going to draw the microtubules. So here goes my microtubules, and I'm not going to be able to draw nearly as many as I'd like to, but here we go. Should have made that a little bit fatter. So maybe what I will do is I will make it fatter here, so I have more space. So here we go, make that a little fatter. There we go, give myself lots of space there, that's this time. Okay, so remember that these microtubules are found in a 9+2 arrangement. I'm not going to draw all 9+2s, I'm just going to draw a couple of sets. So there we go. Okay, we better label this. I guess I should have kept a different color. Maybe I'll label it in red here. Okay, so these are my microtubules. And remember, it's a 9+2 arrangement. Okay. Now maybe I'll keep with the color of the textbook. I'm going to draw you some motor proteins, those little dynein things. So they kind of look like this. Like I said, they kind of look like little legs. Well, in that case, they look like upside down legs. We'll draw some there. And I'm not going to draw them all. I think that is more than enough. So these are dynein motor proteins. And we've already mentioned this, that these things are powered by ATP. Okay, so what is missing here? I guess we're missing our motion. So we can put that in, and I'll put it in over here. And I'll just show arrows. So we'll say back and forth motion. There we go. And I guess the last thing is, how big are these things? Depends on the cell, and depends on whether you're talking about cilia or a flagellum, right? So a cilia can be pretty small, so I'll write that up here. So size. So cilia can be pretty small, so even is down as 10 micrometers. And a flagellum can be massive, so we're looking at maybe up to about 250 micrometers. So very, very different structure, different motion, powered by a different source, different proteins, same name, and kind of the same function. They're involved in swimming, but again, the swimming motion is very different. So there's my beautiful diagrams. Hopefully you like them. Let's move on. We're going to talk about actin filaments now. Got to find the PowerPoint and share that with you. There we go. Okay, let's talk about actin filaments. So actin filaments are microfilaments. That's what our textbook is using. I've been trying to be consistent with the textbook language, but sometimes they're called actin filaments. You can see I have both written there. You can call them either. I use both terms, obviously. You can see the monomer is actin, and the strands themselves are about 7 nanometers. So I'll fill that in to our Word document here, and we'll come back to that in a minute. So maybe I will put a note that microfilaments are also sometimes called actin filaments. There we go. And what are the subunits? The subunits are called actin. There we go. Notice it's in red because actin filaments are usually stained red by fluorescence microscopy. So these ones are the smallest. 7 nanometers is the size of an actin filament, a fiber, or 7 nanometers in diameter. Okay, we'll come back to this once we've got a chance to see what these things are doing. There's a picture of some actin filaments. You can kind of think of it as a little bit like a rope, right? You have 2 strands and they're intertwined within one another. You may notice that it's talking about where they're found here. They're kind of like a web found beneath the plasma membrane. So they're pretty close to the plasma membrane. So they're going to do a lot of things that have to do with interacting with the cell surface, such as changing the shape of the membrane and things like that. So let's take a look. I have some other pictures. There's a nice little diagram showing the protein fibers and all the proteins kind of stapled together there and in the filament. Okay, so let's talk about some of the functions of these things. You may have heard of actin when we're talking about actin and myosin. And where do we find this structure? This structure is in our muscles. So animals have these fibers in their muscles, and you can see from this diagram, there's the actin filaments here, these little orange interwoven fibers. Those are actin filaments. And then we have these bigger filaments that are called myosin filaments. So you're probably wondering what myosin is. Myosin is a motor protein. So they're just very big motor proteins. So what are motor proteins again? They use ATP. So if you take a look here, and you can see it in the video, maybe I'll come back to this video in a moment. I know I have a slide that kind of shows this in action. But what you have is these actin filaments and these myosin filaments, and they slide with one another. They ratchet back and forth using ATP when our muscles are moving. So if you take a look at this, this is showing we have relaxed muscle on the top, and they're kind of loose, and the contracted muscle, the fibers get pulled together. So if you take a look at that video that I have on the previous slide, if you watch, that's an actual video of some, is it playing? Let's try press and play. It's an actual video of some muscle fiber, and you can see the fibers kind of wiggling back and forth as they're contracting and relaxing. So pretty cool. I'm not going to go through all the stages of this. I think there's, if you want to read about it more, there's a lot of detail in chapter. It's like 50 or 53 in the textbook, which is more about the physiology of these things. But it talks a lot about the ATP and how exactly that this is done. It's pretty cool stuff. So a second thing that actin filaments are doing is something called amoeboid movement. So amoeboid movement means we're talking about movements of amoeba. So amoeba are single cellular protists, and you can see there's one there, and he's trying to move, and they're moving around by changing the shape of their cell membrane. So these projections they make, I think I pointed these out before, are called pseudopods. So that means fake feet. I guess that's down here at the bottom, I see. And there's some actin filaments there, and some of them are in something called the plasma gel, and those are kind of more solidified, stuck together actin filaments. And we also have some that are in the plasmazole, which are more liquidy. So what they do is they change it. They make it more liquidy or less liquidy to kind of change the shape of the cytoplasm. It's pretty cool. I have a video or two to show you. Here's a video. I actually took this video here a few years ago now, and you can see that that amoeba is changing shape. It's growing out its pseudopods, and it's stretching out. And you know, I know the video is very short. It's only about 10 seconds, but it's moving from the right side of the screen to the left side of the screen. You're going to take a look at some other videos that I took for tomorrow's lab, and you're going to see some other amoebas in action. So let's add to those notes before we forget what they are. So back over here, what did we have? We had muscle contraction. Okay, so not a J and M, muscle contraction. And we just talked about amoeboid movement. So movement of amoeba. Oh, and cellular locations, we can fit that in too. So we'll say beneath the plasma membrane. Back to our PowerPoint here. So what's something else these things can do? In plant cells, sometimes when we're looking at our LOD, if they're really fresh, we can see this. Not often. Often they're just, once they die, they don't, you know, they're not making ATP as much anymore, and you don't necessarily see this. But what's going on here? If you look carefully, you can see chloroplasts are moving. So why are they moving in chloroplasts? Well, you know, plants are all about diffusion, right? They want water to diffuse, they want oxygen to diffuse out, and carbon dioxide to diffuse in. So they're moving, they're literally moving their chloroplasts around to kind of get everything just moving a little faster. So you can see that in this diagram, it kind of looks like they're moving around the outside, which they are, because on the inside of the cell is the cell vacuole, and there's no space for the chloroplast to move there. So there's one other thing. So you may have noticed a lot of those actin functions were specialized for specialized cell types. Here's something that's specialized to all animal cells. Is the formation of the cleavage furrow. So we'll talk about this a little bit later when we talk about cell division, but when plant or animal cells divide, the last step is to pinch the cytoplasm, and this is part of cytokinesis. So how do you do that? Well, we have this cytoskeleton network just beneath the membrane, and so it's basically grabbing onto the membrane and pinching it to make two daughter cells. Okay, so let's complete our notes. There are other functions of all these cytoskeleton areas, but these are the ones that you should know for this class. So what did we have here? We had cytoplasmic streaming, not streaking. Okay, and this is in plant cells. And we have formation of a cleavage furrow, and this is animal cells. There we go. Okay, moving on. I know we got 10 minutes left. We are doing just well for fine. We just need to talk about intermediate filaments, and that one's a little shorter than the other ones. So back to our PowerPoint. Let's talk about those intermediate filaments. So you can see intermediate filaments are somewhere in the middle, 8 to 12 nanometers. And what are they made of? They are a little bit more diverse. A lot of the proteins are called keratin, and some of the proteins are called keratin-like proteins. So I'll put that down in our PowerPoint right now. And we've already talked about one type already, and I'll get to that in a moment. So intermediate filaments. And we have keratin and keratin-like proteins, such as lamins. So where did we see that word before, lamins? The nuclear lamina, where we saw them. So I think I have a picture showing that in a moment. So what did we say, 8 to 12 nanometers, and there's no word for medium-ish, medium-ist, so I'll just say medium. Don't think we can say medium-ist, but they are in the middle. And where are they found? They're found in all sorts of specialized cells. We'll talk about that in a moment, such as we already talked about the microvilli in intestinal cells, and also found in the nucleus, or nuclear lamina. And of course, that kind of fills into the structure too. But let's take a look at some pictures here. All right, back to the PowerPoint. I just got to find it here. Okay, there we go. There's the PowerPoint. So there's our microvilli, right? So one of them is a microvillus. So notice that the microvillus or villi are made of both microfilaments and actin filaments. So maybe I should add this to our microfilaments list. So as I mentioned, these are found in all sorts of specialized cells. So think about all the cells in your body. There's many shapes and sizes. Anything with a funny shape. So like a neuron is it's a long, skinny cell. There are intermediate filaments that are there fulfilling that role to give it a certain type of structure. These tend to be a little bit more permanent. They're not moving things around like actin and myosin. The only case where things are broken down for intermediate filaments, of course, are the nuclear lamina, which are broken down during mitosis and meiosis. So I thought I had a picture. Yes, there we go. There's our nuclear lamina. And those are the fibrous structures on the inside of the nuclear membrane. Okay, so let's write our last notes here for this table that we've been filling in. Just going to get back to actin filaments, and I'm going to write that last one, which is microvilli structure. And we can add that same one to the intermediate filaments. So microvilli structure. There we go. And I'm just going to put nuclear lamina. So nuclear lamina. There are lots of other examples. I'm just going to leave you with 2 examples for the intermediate filaments. Okay, so hopefully you got all that. I know this table is a little bit different from the one that's in your notes, but that's okay. I'm sure you'll figure it out. So a couple other things to say about the cytoskeleton. I know it has a lot of weird words. Okay, this is unfortunately, what cell biology is kind of like. Some of the words are familiar to you. Nucleus is one of those words you learned a long time ago. And this is a very specialized area of cell biology, and it is becoming really important. The amount of people that are studying the cytoskeleton has just exploded. There's entire journals devoted to the cytoskeleton. Here's one here called Traffic, International Journal of Intercellular Transport, because we're trying to understand, how cells work. What's going on. Things are moving around, and there's a lot of implications to diseases. Huntington's, for example, there's a protein that moves around between the nucleus and the cytoplasm, and trying to understand exactly how that works. So very, very important stuff. And so I encourage you to learn a little bit about them, even if there are lots of strange words. So the last thing, kind of the footnote to this particular unit, is that I think our textbook has finally stopped saying this, but up until I think the most recent edition, it said, cytoskeletons are found in eukaryotes. Great. So what about bacteria? It used to be thought that bacteria did not have cytoskeletons. We kind of known for about 15, maybe a few more than 15 years now, we've been discovering these structures in bacteria that are very much like cytoskeletons. So this protein here, there's a geneticist named that. That's a wonderful name, MREB. That is actually an actin homolog. What do I mean by actin homolog? I mean it's very similar to actin in how it folds and looks, just not similar in enough that you would see it through a search in a genetic database. You'd have to really look for it. And notice this MREB, this actin homolog, is kind of making kind of a twirly structure here, right? And so this cell owes its shape to this MREB protein. There are other bacterial cytoskeletal components. There are things that look like tubulin. There are things that look like intermediate filaments. You can see the microtubules are involved in cell division, so they're pinching off a cell membrane. Again, very similar to a function that you see in a eukaryotic cytoskeleton. Okay, so I think that is it. That is all for the cytoskeleton. So done a couple minutes early. So maybe you'll use those last few minutes to, do some tweaks to your lab report. So I know you're looking forward to getting it done, and I will have those midterms graded hopefully soon, because I really don't want more grading. I want to at least get rid of one pile before I receive the next pile, which is your lab reports. So I would encourage you maybe by the end of the day tomorrow, check Moodle to see if your exam grades are there. If they're done, I'll have the exam grades posted to Moodle. So have a great Wednesday, and see you on Friday.